Hello and welcome to BiochemSerye, or the Biochemistry lesson series. This series is entitled The Basics of Chemistry. It is important that we review some basic concepts of chemistry before dive straight onto studying the much more complicated world of biochemistry. This series is composed of 6 lessons. Please feel free to subscribe to be updated of the latest lesson installments for this series. This is the 3rd episode of the Basics of Chemistry lessons series and includes Lesson 5. Please click on the card above if you want to see all the videos in the Basics of Chemistry playlist. Lesson 5 discusses the functional groups that comprise the biomolecules. We will discuss Lesson 6, the concept of chemical reactions, in the 6th and last lesson of this playlist. The succeeding lessons are in the subsequent videos. Please subscribe and click on the notification bell to be notified of the succeeding lesson uploads. It is best to discuss functional groups for us to understand the essence of biomolecules in biochemistry. The key element is carbon, which forms 4 covalent bonds with other atoms. Carbon atoms are joined through double or single bonds to form the carbon backbone for structures of varying size and complexity, as we can see here. Groups containing 1, 2, 3, 4, and 5 carbons plus hydrogen are referred to as methyl, ethyl, propionyl, butyl, and pentenyl groups, respectively. Look at this closely. If the carbon chain is branched, the prefix iso is used. If the compound contains a double bond, ene is sometimes incorporated into the name. Carbon structures that are straight or branched with single or double bonds, but do not contain a ring, are called aliphatic. However, carbon-containing rings are found in several biologic compounds. One of the most common is the 6-membered carbon-containing benzene ring, sometimes called a phenyl group. This ring has 3 double bonds, but the electrons are shared equally by all 6 carbons and delocalized in planes above and below the ring. Compounds containing the benzene ring, or a similar ring structure with benzene-like properties, are called aromatic. This illustration might look daunting, but what we see here are the basic building blocks of the different biomolecules, the functional groups. To start, it is best to understand that biochemical molecules are defined both by their carbon skeleton and by structures called functional groups that usually involve bonds between carbon and oxygen, carbon and nitrogen, carbon and sulfur, or carbon and phosphate groups. As we can see here, there are carbon-oxygen groups, carbon-sulfur groups, carbon-nitrogen groups, and the sulfur and phosphorus-containing groups, the esters and the amides, respectively. In carbon, carbon and carbon, hydrogen bonds, the electrons are shared equally between atoms, and the bonds, as we have learned in a previous lesson, are nonpolar and relatively unreactive. And we have learned as well that in carbon-oxygen and carbon-nitrogen bonds, the electrons are shared unequally, and the bonds are polar and more reactive. Thus, the properties of the functional groups usually determine the types of reactions that occur and the physiologic role of the molecule. Functional group names are often incorporated into the common name of a compound. For example, a ketone might have a name that ends in -one, such as acetone, and the name of a compound that contains a hydroxyl, like an alcohol or O group, might end in -ol, as in ethanol. The carbon, carbon, and carbon, oxygen groups are described as oxidized or reduced according to the number of electrons around the carbon atom. The carbon becomes progressively more oxidized and less reduced as one goes from an alcohol to an aldehyde or a ketone to a carboxyl group, as shown here. Carbon-carbon double bonds are more oxidized and less reduced than carbon-carbon single bonds. On the other hand, the acyl group is the portion of the molecule that provides the carbonyl, single bond carbon double bond oxygen group in an ester or amide linkage. It is denoted in a name by a -nyl ending. For example, the fat stores of the body are triacylglycerols. 3-acyl or fatty acid groups are esterified to glycerol, a compound that contains 3 alcohol groups. Moving on, let's look at functional groups that are acidic. Chemically, acidic groups are defined as groups that contain a proton that can dissociate, which will usually leave the remainder of the molecule an anion with a negative charge. In biomolecules, the major anionic substituents are carboxylate groups, phosphate groups, or sulfate groups, as shown here. They are sometimes suffixed with the term ate to denote a negative charge. Additionally, phosphate groups attached to metabolites are often abbreviated as P with a circle around it, or just as P, as in glucose 6P for glucose 6-phosphate. On the other hand, compounds that contain nitrogen are usually basic and can acquire a positive charge as seen here in this slide. Nitrogen has 5 electrons in its valence shell. If only 3 of these electrons form covalent bonds with other atoms, the nitrogen has no charge. If the remaining 2 electrons form a bond with a hydrogen ion or a carbon atom, the nitrogen carries a positive charge. Amines consist of nitrogen attached through single bonds to hydrogen atoms and to 1 or more carbon atoms. Primary amines, such as dopamine, have 1 carbon-nitrogen bond. These amines are weak acids with a pKi of approximately 9, so that at pH 7.4 they carry a positive charge. Secondary, tertiary, and quaternary amines have 2, 3, and 4 nitrogen-carbon bonds respectively. From another perspective, we can look at the different functional groups as to how they are produced. As we can see here in this illustration, most biomolecules are derivatives of simple compounds of the nonmetals oxygen, hydrogen, nitrogen, sulfur, and phosphorus. The biochemically important oxygen, nitrogen, and sulfur compounds can be formally derived from their compounds with hydrogen, for example H2O in water, NH3 or ammonia, and H2S or hydrogen sulfide. In biological systems, phosphorus is found almost exclusively in derivatives of phosphoric acid, H3PO4. If 1 or more of the hydrogen atoms of a nonmetal hydride are replaced formally with another group, R, for example, alkyl residues, then derived compounds are obtained. In this way, alcohols and ethers are derived from water. Primary, secondary, and tertiary amines are obtained from ammonia. The thiols and thioethers arise from hydrogen sulfide. New functional groups can arise as a result of oxidation of the compounds we just enumerated. For example, the oxidation of a thiol yields a disulfide or RSSR. Double oxidation of a primary alcohol gives rise initially to an aldehyde, and then to a carboxylic acid. Feel free to pause this presentation to have a longer time to check out the illustration shown here. In contrast, the oxidation of a secondary alcohol yields a ketone. The carbonyl group, C-double-bond-O, is characteristic of aldehydes and ketones. The addition of an alcohol to the carbonyl group of an aldehyde yields a hemiacetal ROCHOHR. The cyclic forms of sugars are well-known examples of hemiacetals. The oxidation of hemiacetals produces carboxylic acid esters. Moving on, it is worthwhile to know that very important compounds are the carboxylic acids and their derivatives, which can be formally obtained by exchanging the OH group for another group. In fact, derivatives of this type are formed by nucleophilic substitutions of activated intermediate compounds and the release of water. Carboxylic acid esters, ROCOR, arise from carboxylic acids and alcohols. This group includes the fats, for example. Similarly, a carboxylic acid and a thiol yield a thioester, RSCOR. Thioesters play an extremely important role in carboxylic acid metabolism. The best known compound of this type is acetyl coenzyme A. Carboxylic acids and primary amines react to form carboxylic acid amides. The amino acid constituents of peptides and proteins are linked by carboxylic acid amide bonds, which are therefore also known as peptide bonds. To continue, phosphoric acid, H3PO4, is a tribasic or 3-product acid. That is, it contains 3 hydroxyl groups able to donate hydrogen ions. At least one of these 3 groups is fully dissociated under normal physiological conditions, while the other 2 can react with alcohols. The resulting products are phosphoric acid monoesters and diesters. Phosphoric acid monoesters are found in carbohydrate metabolism, for example, whereas phosphoric acid diester bonds occur in phospholipids and nucleic acids. Compounds of one acid with another are referred to as acid anhydrides. A particularly large amount of energy is required for the formation of an acid anhydride bond. Phosphoric anhydride bonds therefore play a central role in the storage and release of chemical energy in the cell. Mixed anhydrides between carboxylic acids and phosphoric acid are also very important energy-rich metabolites in cellular metabolism, as we see in adenosine triphosphate or ATP. This concludes the 5th lesson in the Basics of Chemistry lecture series. The next episode, Chemical Reactions, is featured in the next video. Thank you very much. Thank you for watching this episode in this playlist. Check out the other videos in this playlist, and please do subscribe to our channel and click the bell to be notified of new video episodes that will be regularly uploaded.